only when I give tests am I talking.
I have all forms.
Alright, the easy one. Know what the term pathogens mean. What a decomposer does. Difference between prokaryotic cells, eukaryotic cells, and acellular organisms. Acellular. Remember chapter 1 where we classified all of those different 6 main groups, whether they were pro, U, or acellular. Make sure that you can do that as well. Make sure you know some of the basic characteristics of viruses that we talked about. Like you gotta have an electron microscope. Very small. They do not have all of the components it takes to be a cell. But now they still have DNA or RNA, so keep that in mind. The Swan-Necht class experiment. So that goes into making sure you know what spontaneous generation is. Yes. So make sure all those little scientists that I pointed out, I think there was like maybe 4 or 5 in that chapter 1, make sure you know what, who did what, who was responsible for it. Yes, just the ones on the PowerPoint. There's a lot of people on spontaneous generation, so I just gave you one for the end. There's actually a lot of people working on that. So. Anyway. No. Parts of the scientific method, did I say that yet? Cox postulates. Taxonomy. Make sure you know the hierarchy. Remember that. You play chess or fine games. So make sure you look through all of those taxonomy slides. I don't ask you the different keywords there. So don't worry about knowing the different keywords. I didn't ask you. But I do ask you the hierarchy as far as what group is the most specific, which group is the largest. So remember those domains are the largest group, and then it goes all the way down to narrowing it down. Largest to smallest. But yeah, it is in the order from largest group to smallest group. A couple of taxonomy questions, so just pay close attention to those slides. How to write a scientific method. Take a look at the, wow.
We get crazy in here.
Yeah, that's right. Also about really cold.
Mm-hmm.
You guys know what sterile means, and make sure you know. Yeah, I have a couple of these guys on different sciences. All right, know the structure of an atom. Make sure you know things like atomic number, mass number, what an isotope is. along with the structure of the atom, I mean, you know where everything's at, and make sure that you remember this rule to the first 3 shells as far as how many electrons fit. A question about polarity. pH. You want to know a description of covalent ionic hydrogen bonds. Then when we get into those 4 macromolecules, carbohydrates, lipids, proteins, nucleic acids, make sure you know all the examples I gave you and their structure. Examples, structure. There are a lot of those questions. Yes. I'm multiple choice. This whole thing is all multiple choice. Yeah. Oh, age of that. Age of that. Macromolecules. Now let's just go to school. You know, they got crazy with them. The 5 I's that we just went through today. Classification of media and different types. Make sure that you would know the appearance in rock versus semi-solid versus solid. How much agar is in each, or is not. Pure culture versus a mixed culture, what's the difference? Those 3 methods for obtaining isolated colonies to streak before spread. Make sure you pay special attention to that mannitol salt property. And the microscopes, I want you to know bright field, dark field, phase, fluorescence, and electron. Bright field, dark field, phase, fluorescence, electron. That chart in the book that I had you start, that had all of those different types of which ones are positive stains, which ones are negative stains. Look to see the ones that fall into what category, things that they have in common. Also, the different dyes. So what are positive stains, what are negative stains? So you want to know the PowerPoint, you want to know the chart. So make sure you know the organism has a negative charge on the outside, so those positive dyes will find too. But then make sure you know the examples names of positive dyes, as well as the negative ones. And the negatives find everything else around it. Why? Not that.
Negative charge on outside and positive.
Yeah, the organism has a negative charge on the outside, which microorganisms do, and a positive stain will find to it. So that negative stain stains everything else but the microorganisms. So kind of eliminates what you want to see with the outside. You guys can We can get out your loop sterilizer, maybe it'll warm it up. The loop sterilizer, there's a little hair dryer with some things underneath that. And then are there any questions on the review? Well, you got it.